Morning, everybody, and welcome to Grand Rounds in the time of COVID, which means virtual. Next week, our Grand Rounds speaker is going to be Dr. Lisa Marsh, who's from Dartmouth, and will be speaking on something very relevant to the current situation, which is digital therapeutics for behavioral health, anytime, anywhere healthcare. So no announcements for me to make other than the fact that we continue to wend our way through this COVID pandemic. The actual infection rates have plateaued and are declining very gradually, but that's good. And with the change of seasons, presumably it will continue. With the vaccination, it'll continue. The thing is that we can't relax the infection control or protection guidelines, masks, distancing, disinfection, all of that needs to be maintained in order to accelerate the dissipation of the virus. And also particularly with the emergence of new strains that seem to be responding, or the vaccine seems to be covering, but do have some effects in terms of the degree of immunity conferred. So we're not out of the woods yet, but things are moving in the right direction. And the hope is by late summer, fall, things will be substantially better, but we have to really remain disciplined. Having said that, I want to turn to introducing our Grand Round speaker for today, who represents a real pleasure in a number of ways. First, he's somebody who actually began his career at Columbia. Second, he was in a very prominent role that controlled many of your lives, even if you didn't know it, when he was at the NIMH. And then recently returned to New York when he was recruited by Tom Maniatis at the New York Genome Center, and immediately we took the opportunity to engage him and nominate him for a faculty appointment in our department, which he's accepted and is now co-directing with Stephen Kushner, our genetics interest group. And that's none other than Thomas Lehner. So Dr. Lehner is a native of Austria. He graduated and received his PhD in biology from the University of Vienna. And then he was a postdoctoral fellow at Columbia. And he was mentored by none other than the great Jörg Ott, who some of us old timers may remember, but was really one of the leading figures of the era in psychiatric genetics, and also had the kind of foresight of having an emphasis not so much on the gene jockeying sequencing, but on the computational aspects of it. Then Thomas moved with Jörg to the Rockefeller University, where he was in various positions for several years until he went off into the private sector in the year 2000 and founded a company which then got acquired by Millennium, which is a major genetic sequencing company in the precision medicine space, and was director of science and research for Millennium Pharmaceuticals. But in 2004, he was recruited to the NIH and became the chief of the genetics branch of mental disorders, and ascended to a higher position where he was chief of genomics research branch within the division of neuroscience and basic behavioral science at the NIMH. And that was the era when he governed the policies and the funding, or the research portfolio in psychiatric genetics for the Institute that many of us have relied on or benefited from. In that capacity, Thomas, when you're on that side of the table, giving grants as opposed to applying for them, you have to establish sort of a strategy, a mission, a policy, a set of priorities. And in his tenure at the NIMH, he developed 30 NIMH research initiatives and was the NIMH lead on 6 trans-NIH research initiatives. And even though individuals in those positions don't necessarily get the attention, glory, credit. They're kind of the unsung heroes or villains if they, come up with clunky ideas. But in Thomas's case, he really, I think, drove the genetics agenda for mental illnesses and psychiatric brain disorders for a considerable period of time and has a tremendous legacy as a result. His publications, sort of indicate the breadth, and this may date him, but in a good way, in the sense that his first publications, let me see if I can find it in his multi-volume CV. His first publication was with Yergat, and it was on linkage analysis, if the young ones can remember what that is, for a disease with complex mode of inheritance. And in his second paper, his co-authors were Chuck Kaufman, who was a Columbia genetics researcher, and Conrad Gillum, who led the Genome Center at Columbia for a long period of time before he went to the University of Chicago. And then his next paper was with Linda Brestowitz, and then with Jim Knowles. And his early papers are with all the genetic luminaries who began at Columbia and then left. But we're not going to let that happen again. And then, he had papers in the course of his tenure at the NIMH in collaboration with some of our faculty. He collaborated with John Mann and Vicky Arango on endophenotype and genetic studies of suicide. He had a paper he co-authored with Mario Kendo on genetic repositories for the study of psychiatric disorders in ethnic minority populations. And just to show you that he hasn't lost his touch and become just a paper pusher, his most recent publications in 2021 include a publication in Nature Methods on quantum computing at the frontiers of biological sciences. And then also in collaboration with Myrna Weissman, he has epidemiologic genetic studies of disorders in Africa, showing that the global mental health issue and the need to be able to extend our reach to developing and really underserved countries is important. So it's great to have Thomas back in New York and at Columbia and in collaboration with the Genome Center. And we look forward to his participation in many things and his talk today. So welcome, please join me in welcome virtually Dr. Thomas Lehner.
Thank you, Jeff, for this very, very kind introduction. Way too kind, thank you. And it did take me back and and I had to chuckle to myself when I thought about all, the fun, the excitement, and some of disappointments I experienced all these many, many decades ago. As Jeff mentioned, I started my life in the US and in science, right? All these more than 30 years ago. And I spent a good time of my career at the National Institutes of Mental Health. And, from my various experiences that Jeff so kindly outlined, I'll share my thoughts on where psychiatric genetics and genomics as a field is going now, and have some ideas about future opportunities. And the way I've structured my talk is I want to first provide some context for the importance of studying the biological underpinnings of psychiatric disorders. I want to talk a little about the forces behind that current era of progress. I'll provide a summary of what we know about the genetic architecture of two major diseases in that space: autism spectrum disorders and schizophrenia. And I want to briefly describe one of the major existing initiatives that attempts to close the gap between gene discovery and biology, one of the main unresolved questions in our field. And want to end with some thoughts about the possible future that sort of melds basic discovery science with precision, what I call precision psychiatry. So, as neuropsychiatric disorders are disorders of the brain, right? They affect cognition and behavior. they outclaim they have no discernible manifest biological marker, and they are all diagnosed largely by scale-based interviews and lead to categorical diagnosis. And what we know that most onset between childhood and early adulthood. They have huge economic impact, and is depicted here in DALYs, or disability adjusted life years. This is true for the US, where together with neurological disorders, they are in an unenviable first place, and globally, where they are ranked behind cancer. I mean, indeed, data here are a few years old, but I understand it has not fundamentally changed.
I'm sorry to interrupt, but can you share your screen, because we're actually not seeing your slides yet.
Oh, I'm not seeing my slides.
Sorry about that.
Okay, sorry about that. And I practiced, and still I'm not managing, so hold on.
Perfect, now we can see them, thank you.
Okay. So let me go into. So, these are the slides that you have missed then. So this goes back to really description of some of the major categories and the ages of onset. These are the DALYs worldwide, lost to illness. Again, where neuropsychiatric disorders and neurological disorders, are on top. And this slide shows of the disease burden for the US. And all this, I know for many of you, this is nothing new, right? But for me as a non-psychiatrist, when I first saw this data, I found them really compelling and also troubling. And although we currently do not have a credible understanding of the underlying biology of these disorders, and there are no physiological measure of disease symptoms, we have known that they have a biological substrate, and that was based on the observation that they are inherited, right? And thus must have a biological substrate. And that observation was done in the seminal study by Gottesman and Shields from the 60s, where they observed that while the risk for schizophrenia in the general population was about 1%, it increased for relatives of patients, and with the highest risk for the closest biological relatives. And while this early work here was done in schizophrenia, today we know from molecular and twin studies that all neuropsychiatric disorders are heritable, and with the heritability ranging from a high of 80% for autism and schizophrenia to maybe 30% for major depressive disorder. And this, interestingly, is very similar for other complex human traits like type 2 diabetes or inherited breast cancer. So why do geneticists study neuropsychiatric disorders? Well, for one, of course, to identify the genes that confer risk and make up the genetic architecture. Certainly the hope to understand their biological function and how these perturbations of the molecular machines of the brain then contribute to the expression of phenotypes. And then hopefully to build causal models of disease, another big challenge for the field. And then the ultimate goal to develop new therapies and cures. And I know I don't have to convince you that this is the challenge of a lifetime, but when I started out 30 years ago, it was felt by many that the genetic architecture of complex brain behavioral phenotypes in general might yield to what was then recently introduced novel statistical methods and molecular probes for positional cloning. Well, today, I know that we're all working on what mathematicians would call a hard problem, right? And since Jeff talked about the past, the late 80s, early 90s, I picked a few examples of the state of the field. This was actually 15 years ago and not 30 years ago. So, and you can, on this one, you can see how, despite all the advances that we made in the 90s and the 80s, this was still very slow going. So this was the state of what I call the state of the art around the middle of the last decade, the last decade, 2005-ish. So this was full of candidate genes, but candidate genes were still dominant. There were a lot of these family-based linkage studies that were very interesting methodologically, but they were rarely replicated. And there was a plethora of this, what I call ill-conceived candidate gene studies that together with environmental associations sort of commanded wide attention, and none of them were replicated. And in the scant 15, 16 years since, we have made enormous progress. And a little bit, I'll touch on the progress today and tell you what I think we have learned. But before I do that, I want to make a brief detour as to why I think we're living in this golden age of gene discovery. And I apologize for the state of this slide. I think it didn't quite survive the transition from PowerPoint to Google Slides, but I think you'll get the idea. So much of the advances, I think, can be traced to a confluence of 4 key developments that I was fortunate to witness and sometimes helped to bring about during my tenure at NIH. There was this general commitment by the scientific community and funders, right, to make the investments to study the human genome through the development of resources. And I'm thinking here the NIH repository and the UK Biobank. And for example, this line here denotes the increase in samples over a stretch of almost 20 years in the NIH repository. And then there were those technological and conceptual advances that made the study of the genome and the evolutionary forces that shape human traits tractable. Just think next-gen sequencing or single-cell sequencing. And what's frequently underappreciated and unloved by the community, this ecosystem of policies and resources, data sharing resources that made access for data and biospecimens possible. And here I'm thinking of the genomic data sharing policy by NIH, or dbGaP as a database that sort of aggregates all the genetic and genomic data that the field generates through programs that are funded by the NIH. And there was also this cultural shift, right, on how research is conducted with the advent of the team science and what I call collaboratories. Here I'm thinking about the PGC, or the Psychiatric Genomics Consortium, or the PsycEncode Consortium, and many others, including H3 Africa. I always thought, though, that the depiction of the explosive growth of the co-publication network in psychiatric genetics captures this increasing importance of the team science approach. You can see here the increase of the scientific output of the last 15 years. It's really well, depicted here. So that convinced me when I first saw this, that we're onto something, and indeed, progress can be attributed to new ways of doing science. And that theme sort of has kept me busy during my long years at the NIH. So, what is it we know today about the genetic architecture of mental disorders and the multiple levels of this biological hierarchy that ultimately lead to observable clinical phenotypes? And here, conceptually, I would say the nodes on this arc of discovery represent levels of analysis that particular subdisciplines of neuroscience typically inhabit, while the arc itself symbolizes the connections between those various levels of biological hierarchy. And if this looks linear, of course, it's not linear, right? We're looking at a systems level problem. And I like the depiction here from a figure from a paper by Matt State and Pat Levitt from some years ago, that sort of thinks of this problem as genetic risk that's conferred to by more than a single gene can affect different phenotypic outcomes. And the perturbations in the germline affect the molecular machines in these various regions of the brain, and then propagate upwards these complex cellular and anatomical structures to cause symptoms. And then there are these additional elements like the genomic background risk or environmental factors that then together with genetic perturbations throw the brain out of its homeostatic equilibrium and into a disease state. And all of this has to be conceptualized against this temporal axis of development. So it's a highly complex system that we're looking at. And before I go into specific examples, I want to briefly summarize just what the field knows, what we know about the genetic architecture of these disorders. So what we have learned is how genetic risk for many complex traits, including neuropsychiatric disorders, is driven by natural selection, and the risk is distributed across the allelic spectrum from common variants that are frequently located in non-coding regions and have individually small risk increases, but in aggregate, much larger genomic background risk. And you've heard of this probably as the polygenic risk score or polygenic risk. To the other end of the allelic spectrum, to rare mutations, structural variation, and private mutations that are often located in exons, the coding region of genes, with elevated but infrequently large risk. And then there's evidence emerging for pleiotropy within and between disorders, and for a common molecular risk for features such as psychosis that span phenotypic classifications. And then a broad realization that no matter how you slice that phenotypic loaf, a large proportion of its genetic variants remains to be discovered. So a lot of what we know, we are really only scratching the surface. This almost eponymous Manhattan plot has become the most iconic image of the modern genetics era. This one here is from the recent PGC, the Psychiatric Consortium Schizophrenia mega-analysis that uses about 70,000 cases of schizophrenia and 250,000 controls. And they identify around 270 risk loci, all in the common variant space. And all the signals above this threshold have reached genome-wide significance after correction for multiple testing. Here, the x-axis plots individual chromosomes, and the y-axis, the significance level. Now, I need to point out, though, these peaks are, and this is very important and often gets confused, right? These peaks are statistical in nature. They do not generally identify underlying genes or genetic elements, and the path between signal to identified genetic gene is not always straightforward. On the contrary, it's a highly complicated, and there are only now are we beginning to make slow progress in this space. I want to stay with schizophrenia. And this is a little complicated, but I find these two slides, these two figures really capture the complexity of the genetic architecture of these complex behavioral traits and what we know about them really well. So the figure on the left here plots variance by allele frequency against magnitude of risk. The blue dots denote all the common risk variants that have been associated with schizophrenia, and those rare genes that have been, the small number of genes that have been fine mapped to a particular locus. In red are the 10 or so genes that have been identified through, the schema analysis. The schema is one of the major consortia. The paper itself is still under review. It's in Medic's archive. But schema identified 10 genes through a very large meta-analysis of exome sequencing data. And the green dots are chromosomal structural variants that do not map to any one gene, but the deletions or duplications of various size. So this really gives you a nice overview of what we know today of schizophrenia, the schizophrenia risk space, genetic risk space, and how it is distributed across the genome and across genetic variation. Now, you may ask yourself then, if this is it for schizophrenia, right? Perhaps the true number of risk genes for schizophrenia is small, and not much of gene-based risk remains to be discovered. Now, the figure on the right from the same paper tries to answer this question. It plots the different classes of mutations in the coding regions for all genes in the genome that are generally mutation intolerant, so the blue dots here, and finds that there, with those 3,000 or so genes, there is risk associated for schizophrenia, right? And you would think that if we take those known schema genes out of those 3,000 genes, and if we already had captured all the genetic variants, the risk would disappear. Well, however, it does not, right? It barely changes. This category here is for particularly damaging genes with particularly damaging mutations. So for those most damaging mutations, risk remains even after you take off those discovered schema genes. And the authors then calculate that there are at least 100 of these risk genes remain to be discovered. So again, we have not nearly resolved the genetic risk architecture for schizophrenia. And as I'll show you, for autism, it's the same for autism. Oops, I'm stuck.
While you're fiddling with that, let me just ask, how do you know that it's not the effect of those genes? I mean, you still have the background. So if there's a background effect, a polygenic background effect on those, rare damaging genes, you understand what I'm trying to do?
But I think even if you account for the polygenic risk, right? And this, I actually think it is accounted for. Even if you account for polygenic risk, you still have residual risk that would. I wasn't part of this analysis, but if I remember correctly, it was accounted for, and they optimized the number of actual exonic variation that is remains after taking out polygenic risk. So I agree, it would not, and I think without those, not accounting for polygenic risk, it's probably more than 100. They would come up with more than 100 genes.
Okay, thank you.
But in general, that's a very interesting finding that had been debated, and in some sense is still being debated, is what is the contribution of common variant risk, individually very small risk increases for the overall genetic variants for a neuropsychiatric behavioral trait. And this debate, I think, is firmly down on that in general, the largest proportion of risk is in the common variant space. But since the individual contributions of common variants are minute, right? We're talking about 1.02 to 0.5 odds increases for a particular risk. And I think that development brought on then the conceptualization of this polygenic background risk, well, I think that did not, if I remember the year correctly, it was 2014, around 2014, that people introduced that polygenic risk to sort of you say, oh, there's something about the genome that sort of impacts negatively the brain. And that is the polygenic risk, right? And then there are those individual genes, mutations in genes that have a large effect size, but combined, they pull off less of the genetic variation than those, maybe 1,000 or many more common variants do. And that translates to autism as well. This is for autism spectrum disorder. This is also a recent analysis of the Psychiatric Genomics Consortium for autism, and uses a much smaller number of cases and controls, and finds 5 statistically significant common variant signals. However, if you now look at the smaller graphs here at the bottom, by performing the joint analysis with schizophrenia, educational attainment, and MDD, the group identified another 7 independent risk loci, and sort of pointing to the to the overlapping genetic architecture between these phenotypes. So, and equally interesting are results from the other end of the allelic spectrum, with an analysis by the Autism Sequencing Consortium. You see, as you see, I mentioned this. I use these acronyms: the PGC, the ASC, and the schema. All these acronyms denote large consortia that sort of combine various genetic and genomic results that have been generated and create collaboratories for joint analysis. So these are the results of the Autism Sequencing Consortium, the ASC, that identified 102 risk genes. And here too, in this a little complicated figure, but very instructive figure, the overlap between disorders can be made visible at the genetic level. This figure on the left here shows tentative genetic heterogeneity between predominantly autism risk genes, this in the rust color, and those that cause autism, but with neurodevelopmental delay, a more severe phenotype in general. And they are in blue here. And the analysis is based on the frequency of deleterious mutations in the overall autism gene set. And strikingly, these genetic differences are also manifest for other phenotypes that are relevant to ASD. Here on the right, showing that subjects with damaging mutations in this predominantly autism and neurodevelopmental genes, the ASD and NDD genes, start walking later and have a lower IQ. So as the genetic architecture of major neuropsychiatric disorders and their interdependencies become increasingly tractable, I want to point out that what we have learned to date applies almost exclusively to populations of European descent. So in other words, neuropsychiatric disease genetics and genomics has a diversity problem. And I thought this paper by Alicia Martin and colleagues illustrates this really well and shows the extent of the exclusion of non-white participants in genetic studies, a really big problem of inequity, but also a scientific problem that needs to be addressed. And we and others have argued, right, that Africa is in many ways the most informative place on earth to discover the genetic basis of complex traits, because Africa harbors more human genetic variation than anywhere else on the planet. Though, one obstacle to studying mental disorders on the continent that needs to be addressed as well is how little has has been done today to understand the epidemiology. This figure is from a recent scoping review by Myrna Weissman's group, and that was work that, as Jeff mentioned, I was involved in as well. And it sort of crystallizes that while genetic epidemiology and gene discovery in ancestral populations are clearly areas that are ripe for improvement in investments, fortunately now funders are beginning to address this. Right? NIMH recently announced an RFA that focused on human ancestral genomes and found a large consortium and funded, among others, a large consortium with collaborators in Africa, in several countries, at the Broad, UCLA, and here at Columbia, that will generate at least 150,000 whole genomes in patients with psychiatric disorders. So the Neuropsychiatric Gene Discovery Enterprise project has led to the identification of a number of genes, and pathway and other analysis shown that they map, these are regulatory elements. They're often associated with ion channels, synaptic function, gene expression regulations, and a few other pathways. So now we have this repertoire of genes that sort of opens up an avenue for further functional work. And for this, I want to briefly mention the PsycEncode Consortium. It's one of the major initiatives in this space. And PsycEncode set out to understand how germline risk perturbs the molecular machinery of the brain and to identify non-coding elements and their role in disease. And as with all of the NIH initiatives that I was involved in, also to create a resource for the community. PsycEncode started with 3 themes, the developing brain, the adult brain, and one that was disorder focused. The phase 1 that recently ended was done mostly about tissue and in the prefrontal cortex. This was all driven by limited resources, but now phase 2 has been expanded into single cell profiling and recently was ramped up with another round of funding. And to date, that particular effort includes almost 3,000 brains across several disease areas and has yielded a number of papers, including special editions, Science and Science Translational Medicine. I just want to give you a brief example. One of their original capstone projects that I found particularly interesting, where they integrated transcriptomic data that were generated by the PsycEncode Consortium and data that was available from other resources, and make some interesting findings, including the role of splicing and isoforms that had previously was previously unrecognized, and their role in the biology of this disorder. So that was sort of a really interesting finding that has resonated with the PsycEncode Consortium members, also with NIH. I think there was anyway, so this is one of the findings that helped make the argument that this space that funders need to continue to make investments in this space. And just here, a plug for the consortium, PsycEncode under NIH data sharing rules, their data are available and accessible as a resource, and are continuously updated as new data are generated. So another important tool in that arsenal that will help delineate the biology of neuropsychiatric diseases are cellular model systems, and I think they deserve a brief mention. So conceptually, a sample is taken from a patient, reprogrammed as a cell from the affected tissue, neurons in our case, and they can then be deployed as a screening assay for drug development, or more aspirationally through the application of genomic technologies, become a treatment modality themselves. And here's an example of this approach from the Brennan lab. Kristen's work shows how deletions in the orexin gene in iPS neurons from patients affect isoform levels and associated cellular phenotypes. So really you can do cellular phenotyping, and this could potentially be used as an assay for pharmaceutical screening. So as we make progress, along this arc of discovery, and here we mapped this approach to the major existing research consortia that I've been mentioning, and all their interdependencies. And you may recognize some of the acronyms, as I talked before, PGC, Psychiatry, ASC, and others. I touched on some of these nodes, right, like gene discovery, molecular networks, cellular models, but others like circuit level studies, right, and clinical implementation are notably still missing. So what are some of the areas that I think we as a field need to continue to make investments in and pursue? Well, for one, do more of the same. I think gene discovery, particularly in diverse populations, needs to continue to develop a comprehensive understanding of the genetic architecture across human populations. We have only recently scratched the surface of building comprehensive models of disease, and efforts in single-cell genomics are only beginning to take off, so we have ways to go. And how genes and their molecular perturbations causally impact behavior across levels of analysis remains unknown. And finally, I think the implementation of genetics and genomics in clinical medicine is an aspirational goal that we need to achieve in small but considered steps. So creating these causal models of disease across 2 or more levels of molecular hierarchy is of course not straightforward, and may even mean different things to different people, but it will have elements of systems biology, will have a strong computational focus, and span disciplines from inside and outside neuroscience. So break down the silos between the various scientific disciplines and investigators. Now, while I was at NIH, I thought about this quite a bit, and I was able to shepherd a few initiatives through that I now call convergent neuroscience, and including this recent funding opportunity. So finally, extending the arc, right, including translation and clinical practice, and the idea that elements from these various nodes can and should be integrated into clinical care and become part of a learning healthcare system, I think is an ultimate goal. And such a cohort would then integrate diagnostic, demographic, patient care data, genomic data, other biomarkers, and medical records. And there are several existing initiatives to create such a precision psychiatric cohorts, including one here at Columbia. I want to end with an example that I hope will illustrate how such a cohort could be used for translation into a clinical setting. So one of the strongest G signals for schizophrenia is in the major histocompatibility complex on chromosome 6, right, MHC. MHC is a complex region and had proven impenetrable to fine mapping until 2016, when the groups of Steve McCarroll and Beth Stevens were able to map the signal to a gene in the complement system, the complement component 4 gene. And they were also able to show that, at least in mice, C4-mediated synapse elimination during postnatal development. So C4 now mediating synapse elimination well, this finding is hypothesized may help explain the reduced number of synapses in the brains of schizophrenics, right, individuals with schizophrenia. So Perlis and colleagues picked up on this and added strength to this argument by showing increased C4 dependent synapse elimination in patients. Now they went into patients, from mice into patients, and they developed cellular models. They also showed, and here it gets really interesting for me, that the antibiotic minoxidil inhibits the elimination of synaptic structures. So these are tetracyclines, minocycline or doxycycline, are commonly prescribed antibiotics to treat acne in puberty. They then go to 2 large academic medical centers with 10-year follow-up data and schizophrenia, and analyze electronic medical records for prescription and data and schizophrenia diagnosis. What they find, and this is not showed very well, but it's significant, they find a significant decline in schizophrenia risk among subjects that were prescribed one of those antibiotics for at least 90 days between the ages of 10. And of course, I know this is just a proof of principle analysis with many caveats and not really a true example for precision psychiatry, but it does trace how different levels of analysis can be synthesized into testable hypotheses and be put into real-world clinical settings. And with that, I want to end here. Thank you. And take questions.
Thomas, that was brilliant. I mean, that was what are you doing next week at this time? No, that was a fantastic overview of areas, but what was brilliant about it was the way you could kind of synthesize it and explain it in a way that was understandable to end users like myself and people who are not steeped in molecular genetics particularly. So I just want to ask one question, and then Jonathan is going to moderate the question and discussions. You mentioned on one hand that early slide that if you look at this, you might think that the genetics can be attributed to a relatively limited number of genes and should be therefore understandable. And then you show these slides, 108 genes and 200 genes, and the ones that are GWAS studies with common variants with low penetrance. But in any event, it's a very complex process, whether it's rare mutations against a polygenic background or the missense or loss of function or truncating. Is the genetics of mental disorders so complex because it's the brain and the brain is so complex, or all polygenic medical disorders, whether it's essential hypertension or diabetes or ASHD, comparably complex?
So my cop-out answer is all complex traits are complex. That's why they're complex. But I think the behavioral traits, because of how evolution has buffered the brain and made it more interdependent on other organs, and we know very little about brain function really, gives us an additional level of complexity for the brain. Now, if you think about it, there has to be a lot of redundancy built in, right, so that the homeostasis is maintained. So if you then construct an experiment that will try to help you identify genetic signals, say, at the genetic or at the genomic space, there's a lot of redundancy built into the system, and it's very difficult to push through the what I call the noise of insufficient sample size. I think many of these problems can and could be addressed by large sample sizes. And some of the, I took the slide out, but I had a slide where I showed where we calculated with the simulations on how large numbers of subjects would be required for whole genome sequencing, say, to fully cover the rare non-coding space. And we're talking tens of thousands of cases and controls.
So one last question from me. I remember, this may be really obsolete at this point, but I remember early on being informed that for genetics research, that false negatives are worse than false positives in terms of being able to discern signal from noise and so forth. But that notwithstanding, given the heterogeneity that likely exists within our phenomenologically defined diagnostic categories, how much of an impediment is that? And apart from the deep phenotyping and so forth, is there any ways apart from just increasing sample size and throwing huge numbers at the problem that you can recommend?
Yeah, thank you, Jeff. You point the finger on something that I really didn't address. So we are working with phenotypes that are difficult to measure, and they likely have phenotypic components that span what we now the categorical diagnosis we use now. So I think a deeper understanding of, and they used to be called endophenotypes, then they became RDoC, research domain criteria, all of expressing the need to go closer to the biological underpinnings of a particular particular behavioral expression. And I think that remains a challenge for the field. For one reason, because gene discovery worked too well on the crude, if you will, diagnostic criteria we use, right? I mean, schizophrenia, autism works really well. Best diagnosis works really well. But then if you dig deeper and try to understand the genetic architecture of some of these phenotypes, there is work that shows that you can actually pull it apart. I mean, I tried to touch on it when I showed the autism, NDD, and predominantly autism, delay onset for walking, reduced IQ. I think the most progress has been made in autism in that space, and it's because that there is a large number, because of efforts by private funders and NIH, there's a much more refined phenotyping available than for schizophrenia and others. I know I didn't answer your question well, but it's a complicated problem, and it's not until we find biological biomarkers, right, that clearly project to the underlying molecular biology of a disease, the problem is not going away.
So I have a question. About this persistent problem, or the gap between what family-based heritability studies suggest for any given psychiatric disorder and the amount of variance that can be explained through GWAS. And I guess I'm curious to get your thoughts on that. Is that a problem that will sort of attenuate with time as sample sizes continue to increase, or are we more likely to fill in that gap by focusing our energies on G by E interactions?
Well, so I think the answer, my opinion, and this, ask a different geneticist, a different program person, they'll give you a different answer. So I think that there's clearly a sample size problem, right? I mean, you could say, so how many more common variants of schizophrenia do we need, right? Now we have 250. The PGC did an analysis showing that there's a saturation, but how much more of this common genetic variants do you want to peel off, and what does it matter in terms of understanding the biology and in developing treatments? I think that is a very valid question, right? Now, it's good to have that, to be able to pick off the genetic variants, and then also the problem doesn't get easier if you go to the rare variant space, because they're rare, and they're much rarer than we thought originally. So our sample sizes are still large, right? So that, there's that balance, yes. So I would focus, and I think all the major consortia are shifting over to now looking at, we haven't explored the non-coding rare space really, right? Because we don't have the sample sizes. We have some findings that point that the non-coding rare variant space also contributes to the genetic variants of in meaningful ways. So this is something we still, I think, we still need to examine more closely. And I think we should focus on the coding regions of the genome, because it will hopefully help develop better biological models. Now, your question about gene environmental interactions, and I'll defer to the real statistical geneticists among my colleagues, but these are clearly worth examining, but generally you have to have, they don't come by themselves. So to either have a strong environmental risk that sort of overshadows the genetic risk, or you have a strong genetic risk that overshadows the environmental risk. And I haven't seen work that sort of convinces me that you always need to do both, because if you don't, you miss out on important connections. This does not diminish, there's an interest in understanding gene and environment interactions and the contributions of each. But for gene discovery and for environmental risk discovery, I'm not sure that we need to do it unless we have the data. So I remember being involved in a study where we were able to, we had a very large cohort, and we had zip coding, and we had environmental proxies, right? SCS, pollution, and we used these in a comprehensive study, and found really interesting, but nothing came out that was individually, that we didn't see in some form at the individual level.
Yeah, so if I understand correctly, I think what you're suggesting is that the main effect of each should be large enough that we should detect those without having to just see the interaction term. Yeah. So one of our viewers writes in, heterogeneity plagues clinical diagnostic subtypes and may be partly responsible for the difficulty in finding genetic causes of mental illnesses. Has there been effort to re-examine the clinical phenotypes of patients with a specific genetic abnormality and thereby identify a more homogeneous clinical phenotype?
I would say those efforts are all underway by the various groups that I know for all of them. So the schizophrenia, the PGC put out several papers over the last few years that looked at this problem. There's active work going on at the Autism Sequencing Consortium. And of course, there is initiative by NIH called RDoC, Research Domain Criteria, that sort of pursues the same idea.
Another listener writes in, you have laid out the case that complex disorders are the result of complex systems consisting of many genes. How do you think the pharmaceutical industry might adapt to finding new drug targets if single genes / proteins / receptors are likely to be dead ends for drug discovery?
I would put this into hard problem, to the hard problem category. So my experience is that pharmaceuticals have shown, while I was at NIH, we tried to engage pharmaceuticals on multiple time points during my career. And until the very end, we were not successful. Before I left, the AMP program, the Accelerating Medicines program by NIH Central, or coordinated by NIH Central, has put schizophrenia on their list of projects, and that this is a public-private partnership. So there was clearly interest by pharma to occupy that space. But I cannot, but other than that, I do not know how to engage pharma in ways that would change that. Maybe to put it another way, is if you look at the different genes that have been implicated, whether it's the rare ones or the common variants, do they aggregate in certain functional groups?
Yes, they do, but again, yes. So for example, we have the synapse function, right? But for example, I have not been able, it doesn't mean that these are necessarily druggable targets. Just because we find a strong risk factor in a protein truncating mutation in an important gene does not make it druggable. So I've learned that in discussions with people at pharma. So, and I see often that there's this disconnect between what basic researchers, these are excited genomicists, geneticists, they find these really interesting findings, they put them, they put them in a biological context, and then pharma is not interested. I don't know if some of you may have seen the recent talk by Joseph Buxbaum for the Neuropsychiatric disease genomics meeting at the NYGC, where he used an example of a rare disorder that was a fictitious rare disorder that was well known, that he pitched to pharma and said, would you be interested in that? And they said, oh, we're interested. And then it turned out that that fictitious rare disorder was actually a real disorder, and they were not interested.
So another question is from Ralph Wharton, who asks if you're aware of any ongoing twin studies, any data of special interest.
I'm stuttering because there's a large twin study in the Netherlands. And the reason I'm slow to respond is because I don't know what the status is, but Dorrit Boomsma and colleagues has been very active in that space, and I think they're still publishing. That we actually have samples, so there's phenotypic and genetic material available at the NIMH biorepository for the field to study. And then Ken Kendler and colleagues, of course, I think they're also still active in twin research, and there is a twin study whose name escapes me now because my brain is failing old age. That is also very prominent and is still ongoing.
Dr. Meyer Blaberg asks, what genetic mechanisms might explain the differences between males and females in schizophrenia prevalence? And I would actually add to that, given that the biology is present from birth, what are the mechanisms that would explain the delay in the onset of the phenotype?
Yes, so there clearly, well, not clearly, there may be protective factors, but I have to punt on the question because I do not know the answer. It's a fascinating problem, and that got way too little attention for many years. I think that is changing, but I do not have a good answer. I don't have an answer.
The usual thing that's given, apart from any embryological or developmental neurobiological, is it's endocrine or hormone mediated.
Right. That's the answer. Yeah, it's given. But I understand that depending on the brain region, so I understand we had a conversation with a sex difference researcher, and they told us how difficult it is to actually differentiate those effects, the endocrine effects in tissue. And
yeah, I think that's just kind of a very general answer, which verges on a cop-out.
yes.
So there's a question from Anthony Zogby, who writes, wonderful talk. Given the immense polygenicity of schizophrenia, how would you choose which common and rare variant loci to prioritize for functional studies and therapeutic development?
Well, I would, on the rare side, that's relatively easy because for schizophrenia, there are so few, right? On the common side, so there's some convergence emerging between common and rare in the schizophrenia side, so maybe those. but then I would, pathway, and really it's, okay. More biology is needed, more functional work is needed, and the discussions for the need to be around what is the most appropriate functional work, this genes to biology space, that it has been identified as the next major challenge by NIH, and that needs to be considered.
Yeah, you talked about that, the importance of causal analysis across units of analysis. Could you give some examples of promising work in that area?
No.
Okay. Sorry, I didn't mean that one to be such a
no, it's just,
question.
It's not a difficult, it's just, I think this is an area that is waiting. I think it's very difficult, right? How do you develop causal models that cross these hierarchies, right? I mean, right. I was on a call earlier this morning where we talked about, circuit genomics. How do you actually cross causally, mechanistically, how genetic perturbation affects, circuit function? I confess I do not know the answer, but I know there are many people smarter than me who can and should think about these questions. I think causality is a tricky problem. The definition is tricky because I think often when we talk about causality, we still mean associative and correlative, which is not to say that this is not interesting, right? In a sense, much of what we think of precision medicine is probabilistic medicine.
Although I think you've been appropriately cautious about discussing the therapeutic, implications or preferred strategies. The other thing that has come up from what you were describing with the doxycycline work, as well as a conversation that Jonathan and I had with another person from pharma about another condition, is the critical time for the intervention, and how does one begin to consider that in the context of making therapeutic interventions?
Yeah, I mean, and you can imagine. I completely agree, but it's also an additional complexity of the Neuropsychiatric Genomics project, right? Is that we're working on a time axis, the axis of development, that if you miss out sensitive periods, your model gets messed up if you don't take this into account. I mean, one way of as approaching, making claims for causality would be, model systems and rescue experiments, and see how rescue experiments could genomic editing, and see how genomic editing affects particular phenotypes. I would be more confident to claim causality. Now, I understand that there is also a branch of mathematics that deals with causality, and the idea behind this convergent neuroscience initiatives that came out while I was at NIH, I wanted to get these kinds of thinkers on working with neuroscientists, theoretical and applied, to think about what causality really means in that context. And I'm not sure I have succeeded.
Well, you should know that we have a a nascent center for computational psychiatry and neuroscience, which does exactly that, and it utilizes computational approaches, but also sort of extends to the neurotheory, computational neuroscience people, who don't work on data; they work on simulations. But so Guillermo Jorge, Gaurav Patel, a newly recruited PhD from Caltech who will be arriving this summer, and Sean Escola, sort of form the nucleus of that.
Yeah, I talked to Sean this morning. Actually, Sean was one of the people I talked to this morning.
Just don't let him convince you to get into swing dancing. That's his hobby.
Oh, really? Okay.
There's a question from David Forrest, who writes, because there is less genetic diversity, the further populations have migrated from our common African origin. Does that mean that schizophrenia occurring in such populations would be easier to study to find basic genetic causes?
Easier to study in non-African populations?
Correct.
Well, not now, but easier to study just because genetic diversity, while it makes the study more compelling, it also complicated because of the diversity and the sample size. Sample size needed to be larger. And I have a nice example of that. You may have seen about a year ago a paper in Science by Mary Claire King and colleagues and Ezra Sosa on schizophrenia in the Xhosa. It's a South African ancestral group, where they, because of the circumstances of the numbers were relatively small, but they were able through sophisticated methods, statistical, I want to say tricks, I should not say tricks, statistical methods to actually to identify statistically relevant diversity variation. So it is possible. It's more difficult, but it's possible.
I have a question for to maybe, I don't know if this will stop the discussion, but this is sort of the age-old question. So you have two disorders that are highly heritable, schizophrenia and autism. They have low, presumably low fecundity, reproductive capacity. How come their prevalence or their population frequencies don't get diminished and bred out of the population?
Well, I think they're replenished by, so there's clearly balancing selection, and then they're replenished by mutations. So there's this genomic background risk that really works on many phenotypes, obviously, right? And then on the rare, I mean, if you look at many of the 100+ genes in the autism sequencing consortium are de novo, right? And yes, the schema genes are very, very rare, right? Mary Claire almost works on trios, so many of those are de novo. So it's that they're replenished.
So what you're saying is that the common alleles that provide additive risk are so prevalent and distributed in the population that they can't possibly be bred out, and that the mutations which can occur against polygenic backgrounds and are more penetrant, those are being reproduced through de novo mutations.
I think that's a good approach to think, yes.
And this is getting a little far afield. There has been a renewed, but a increased emphasis on autism, where previously it was kind of hiding in plain sight, beginning in the 1990s and through now. And in that context, the population frequency is said to have skyrocketed. And even though Peter, one of our faculty colleagues in sociology, did a wonderful study showing it, I mean, apart from the increased awareness and broadened criteria, it was really social factors that accounted for the epidemic, so to speak. Is that consistent with your thinking?
I would agree with that. I was never, I mean, so surveillance has improved, diagnosis has improved, more resources are in place, but the drastic increase in ASD prevalence, I think is easier explained by what you just mentioned.
Mhm. And finally, for me, with the proliferation of certain pharmacologic substances used, medicinally and recreationally, like cannabis and so forth, and also in Jonathan's area, the inflated use of stimulants for cognitive enhancement, ostensibly treating ADHD that your parents want for their kids. Do you see this affecting the frequency of psychotic disorders?
So I want to defer to the psychiatrist here, but my understanding is that yes, cannabis is a true risk factor for psychosis and subsequent schizophrenia diagnosis. But I also understand that in the studies that are done, cannabis use is corrected for. So I think you are picking up something that's separate from cannabis use. I don't know about the stimulant use and how that could affect incidence and prevalence of these disorders and genetic studies. I mean, notably, the reason I stick with schizophrenia and autism, they were the easy ones, right? The other ADHD, MDD, bipolar, PTSD, and OCD and others, they all have genetic variants, but they're more difficult to analyze for various reasons, which would be another talk. It's complicated.
Well,
So in other words, I understand it is a problem, but I don't think that it will, that the confounding of substance use is so grave that it would create a problem for the genetic analysis.
Oh, no, I don't think that would be the case either. It would probably simply cause phenotypic expression in individuals that may have been sort of subclinical.
Tip them over, right? Yeah. And this may be a different underlying propensity, right? This could be around resilience. There could be a biological resilience, genetic architecture. I always wanted NIA to study these kind of things, but there was never, I think I did one initiative with NIA as a co-sponsor. We looked at comorbidities, and there was just no interest by that institute to invest in that space.
Okay, I think we've asked you all the questions that we have. So I just wanted to echo what Jeff said, that was a remarkably clear talk given the complexity of the material, and also really both inspiring about what genetics holds for the future, but with a good deal of caution. So thank you so much for your time, and I'll turn it over to Dr. Lieberman for final words.
Just thanks, Tom, and look forward to working with you increasingly in the future. And everybody stay safe, and we'll see you next week, if not before.
Thank you. Thanks. Bye-bye.